Proteins are one of the most important components in our body, and consequently, synthetic biology. In particular, we are going to delve into how scientists design proteins and how the tools and concepts of protein design have evolved over time. Along the way, we will hear from ASU iGEM, whose 2024 project focuses on innovative protein design processes. Let's begin by establishing what exactly proteins are. Depending on your experience, you may associate protein with that thing that you need to eat enough of if you want to build muscle. Or maybe you remember your high school biology class, where you learned about how proteins are one of the major macromolecules alongside nucleic acid and carbohydrates. Either way, both are a great start to understanding the importance of proteins. Proteins are biological structures composed of amino acids. In the human body, only 22 amino acids make up all of our proteins. These amino acids bind together through covalent peptide bonds, creating strings called polypeptides. The exact sequence of amino acids, that is, the type and order, is called the primary structure of a protein. This is the most basic level. This basic order is instrumental in determining the final shape and function of proteins. Once the polypeptides are built, local interactions between molecules cause folding that makes up the protein's larger structure. In the human body alone, there are tens of thousands of different proteins, all with their own unique structure, which allows them to carry out a myriad of tasks.
With so many existing proteins, it can be easy to wonder why it's so important to be able to manipulate and engineer proteins. Why do we need to design any?
People like to say DNA is the building block, but I guess from my perspective, proteins are less well understood and less linearly relatable than DNA, right? Whereas DNA, it's kind of easy to understand when there's promoters, terminators, stop codons. That's like pretty straightforward. Whereas for proteins, there's things like post-translational modifications, different conformations in different pHs, different interactions with other proteins. I think proteins have this versatility that is yet to be unexplored. And it's also like this idea of, you know, things that happen in the background that you don't really think about. I guess my point is proteins really define life.
So the versatility of proteins is what gives them so much potential, and what allows us to engineer them. Since proteins are already serving so many functions almost everywhere in the human body, they are a great vessel for new functions. From creating biosensors to creating new therapies, the possibilities are endless. But how exactly do we take our basic understanding of natural protein structure and design new molecules to meet our goals? One principle used by the iGEM team at ASU is called directed evolution.
We use this system called PACE, which is phage-assisted continuous evolution. And the main idea is you remove one of the genes from the phage so that it needs to infect the bacteria in order to continue proliferating. And inside of that bacteria, you set some kind of condition such that when the phage meets that condition, it can then regenerate itself. So inside the phage, you'll replace one of its vital proteins with your protein of interest. For example, you would replace it with like a T3 or T7 promoter protein. And then in front of your target, so in front of the gene that you removed from the phage, you would actually put like the promoter region, so the intergenic region. This is like a well-documented experiment where they evolved T7 promoters to also target T3 promoters. And the way that mutagenesis happens is actually you include this hypermutational plasmid that has like error-prone polymerases inside of the bacteria. That way your like random mutation rate is much higher than usual.
ASU's project is focused on developing accessible methods for protein design. Directed evolution guides the in vivo side of their project, that is, the experimental side that usually takes place in a lab. But that's only half the story. The other half is called in-silico. How do these two sides of protein design work, and how are they used together?
Currently, our team has identified a set of three, like, uh, how should I say it? They're like these proteins that complex with each other. There's one protein that, well, there are two protein systems. One of the proteins is always a membrane protein. That's the protein that actually excites or undergoes a conformational change in the presence of light. And the other protein is kind of just a kinase. It accepts the excited like phosphoryl group and then starts promoting genetic like transcription. So I think the interesting point about that part of the project is classic pace systems require a lot of complex sugars as a way to like induce the mutational plasmid, induce high stringency or low selectivity conditions. Our approach is to use light. That way it's more tunable, easier to set up, and more, overall just cheaper in the long run.
The associated costs of wet lab protein design is a significant barrier that ASU's project is trying to address.
We're also making like open source designs because to do pace, you also need like a good amount of hardware components and like electrical like wiring components. So we're trying to, our goal is to like make a fully functioning system on the hardware side for under like $200.
On the other side of protein design equations is computation. As you can imagine, even with only 22 amino acids, the number of possible combinations is more than any person could sort through on their own. Computers revolutionize protein design because they boast the capacity to sort through millions of possible combinations.
We started off by assessing the state of the art models that are like freely available with fully like pre-trained without that doesn't require like any like self-training or user training. The idea was to um, basically create a workflow where anybody with little to no machine learning knowledge could generate a binder to a novel interface on a protein, right? So, for example, our team is going to diffuse a binder to these residues on the thumb domain of HIV reverse transcriptase, right? It's been confirmed through literature that this domain like interacts with human enzymes, but it's like no one has ever made a binder to it before. So we're just going to do this from a purely machine learning perspective. We're putting together the models like RF diffusion, protein MPNN to create sequences. We're going to synthesize them and then experimentally validate them with spot arrays. We're also adding on kind of like the machine learning based pseudo binding models. Like there's models like dynamic bind and GAA bind, which will give you kind of like a predicted binding affinity. And we would rank our created binders using the binding affinity rather than the metrics provided by MPNN and RF diffusion. Through our preliminary tests, we have discovered that AlphaFold 2 at least for very small complexes, like the binders that we are creating are very inaccurate. Like the metrics are wildly inaccurate, and they might predict an entire chain of 12 alanines as a helix, even though like biochemically that would never happen. So we're just finding out now that like, oh, these metrics that are given by protein MPNN aren't really the greatest because AlphaFold isn't made for binding prediction, it's made for structural prediction. There's a gap in just peptide-based approaches towards therapeutics in general. Um, I just think that like we have the tools to create just like even a toy model that somebody with just Google Colab free version could run on their own. And I think that's good. We should get people to start thinking about this and start thinking more about peptide binders rather than complex chemical compound ligands.
As mentioned, the predictions made by these programs are not always accurate, and there are certainly gaps in existing tools. Advances in existing technology and practices like those made by the team at ASU contribute to the development of protein design as a whole, allowing for more specific designs and more efficient processes. Overall, the field of protein design is developing rapidly to allow people to design, create, and test proteins that would not exist otherwise. From the basic building blocks available, millions of outcomes are possible. By making experimental techniques more accessible and improving computational models and machine learning, the team at ASU has shown just a few exciting directions within the field.